We are live.
Okay. Clark Christensen, Wesley Butler here for our last genetics video. So let's get into some genetical stuffs. Question 1, part A, has a lot of words. So basically, we made a diagram with 4 lanes. Each one had a different DDNTP, put the reaction mixtures in, and mapped it out. So that's part A. It's that beautiful drawing there for you. You're welcome. B, suppose you use E. coli DNA polymerase 1 during the sequencing reaction instead of Taq polymerase. The E. coli DNA polymerase 1 is not as thermostable as Taq polymerase, so it's just going to denature and the reaction will fail. C, suppose I accidentally add E. coli DNA helicase to the reaction in addition to the normal DNA sequencing reaction components. The reaction will still work because the helicase just separates the strands, which is something that would have happened anyway. D, suppose I add DDATP to one of the tubes and forget to add all of the others to the other tubes. It's kind of janky. So it would work to do the reaction, but your error would make it so that the 3 neglected tubes wouldn't have those chain termination abilities. So the one with the DDATP would be cut into smaller sections, but your other ones wouldn't cut up, so they'd just be these big long things all the same. And that's number 1.
Sweet. Number 2.
We'll see you tomorrow early morning for scouts.
Is pretty much just the vocab.
Later, Harry.
A is the synthesize step and the anneal step, and B is the AMP-R, or the ampicillin resistant marker, and restriction endonucleases recognize and cleave DNA molecules at palindromic sites. C is positive D. So DDNTP lacks the 2' and 3' hydroxyl groups, and E is transformation. F is IPTG is commonly used to induce the expressions of the lac operon, while XGAL is cleaved by the beta-galactosidase to turn the cells blue. That's number 2.
And number 3. You are interested in using PCR to amplify the region of a double-stranded DNA fragment from E. coli. Interested is used generously. So we write the single-stranded DNA sequences that we would use. So here's our answer. There's one of them. There's the other one of them. So for the first one, we got 5' GAGCCTTCACTCAGTC 3'. And for the other one, we got from the 5' direction is CAGTGCAATTGAGGCT 3'. For B, it wanted a mathematical equation for determining annealing temperature. So we did some work. It's beautiful. We also use calculators because it's 2019. So for B, we got 62.375 degrees. And for the second one on it, we got 59.8125 degrees. C, oh, you accidentally add DDCTP. Oh, wait, no. Yeah, DDCTP is accidentally added. So that screws it up because the DDCTP just makes things start chopping. So for the first one, it cuts it real short. So you get 5' GAGC 3'. And for the second one, the very beginning of that primer is a C. So you just get 5' C 3'. The mistake ruined it. And then for D, We learned about DNA-A and DAM, and why are these two proteins not included in PCR? It's because they're not as thermostable. So they in the heat of PCR, they would just end up denaturing and they wouldn't do anything. And that's
Sweet. For number 4, this is our work. And A pretty much asks you to draw the agarose gel electrophoresis, and we drew it according to the table that we're given. B says draw a linear restriction map that indicates the location of those enzymes. And this linear restriction map, so it's the one that's on the bottom here for B. And these could be on either side, but it's going to go in order. There's going to be 50, the BAMH1, and then another 50, and then the equal R1, or 150, and then the equal R1. Then there's going to be 100 base pairs to the HIND3, and then another 100 to the second BAMH1, and then 500 to the end of the segment. And we just figured it out using that chart or that table. D, C, what are the predicted lengths if it's cleaved by all 3 molecules simultaneously? You'd have 150, 50, 2 100 sections, and then a 50 or 500 section.
Yes.
That's number 4.
Number 5 is a Lincoln novel. Here it is, right there. So let me tell you a story. You're going to take some mature mRNA. You're going to take a poly-DT primer. You're going to put those two together, and then you're going to add some reverse transcriptase and some DNTPs. Once you do that, you're then going to add RNase H, and then you're going to add DNA polymerase 1. And once you do all those things, it's going to give you the cloned DNA. Then you're going to take your cloned cDNA. There's a difference. Then you're going to add your restriction enzymes to that, and you're going to drop some plasmids into the tube with it. You're going to allow cleavage by the restriction enzyme.
Good there.
Wait, what?
Good finish there. You're going to allow cleavage by the restriction enzyme.
Yes.
This is genetic stuff.
Yeah, the cDNA. Then you allow the two to connect together, and then you let DNA ligase put some of that in there. The DNA ligase is just going to seal the little chunk into the big chunks that you cut apart, and now you just got one extra big plasmid there. Pretty great. So our plasmid vector, it contains the beta globin gene, and with that, the LAC-Z gene turns on the lac operon, producing beta-galactosidase. And so these cells, all of them that you got there, the ones of ours that have our cDNA that have this beta-globin gene and all that gets turned on, because of the beta-galactosidase, those cells are going to turn blue. So when you blue-white screen them, they'll pop up and you know what's what.
Right on. So number 6, the last one, just says research CRISPR-Cas9 and write your paragraph. I could read you my paragraph, but I'm going to
Please do.
just submit it as group work. So that's it for our video. Last one.